Good afternoon, everyone. Okay, good. My name is Rohit Bhargav, and I'm from your host institution, University of Pittsburgh. I'm a pathologist. I direct a lab at UPMC McGee Women's Hospital, which is our flagship women's hospital where we always process and report on all breast pathology specimen. Really excited to be here and really excited to see all scientists, advocates, and clinicians under one roof with a common goal. I want to thank Dr. Schnitt for a wonderful lecture, which provides us a nice backdrop of what we are going to discuss next in this session. We are going to discuss further the pathology of ILC and mixed ductal and lobular carcinoma and how E-cadherin inactivation shapes tumor microenvironment. Although the focus of this symposium is really on invasive, but we also need to know where invasive carcinomas come from. So we'll also touch base on LCIS, which Dr. King will be discussing. But first up, we have Dr. Maxime D. Shepard, who will be discussing homogenization of pathological diagnosis of ILC. Dr. Shepard works at the Laboratory for Translational Breast Cancer Research, Department of Oncology at the Catholic University in Leuven, Belgium. And it seems like he hasn't made it to Pittsburgh, but he will be doing it from Zoom. So I think we have to make him a co-host. He should be good. Dr. Shepard.
So thank you very much for this kind introduction, and thank you very much to the organizers for letting me present this work online. I was really looking forward to be there in person, but unfortunately the coronavirus decided differently. So today I'm going to present work that has been done from the ELBCC Pathology Working Group in order to improve the diagnosis of ILC in the pathology lab. So as Professor Smith already highlighted...
Dr. Shepard, could you please speak up a little?
Okay. So as Professor Smith already highlighted, ILC is a pathological diagnosis. And when we look in the WHO Blue Book, we see that indeed it is essential to recognize some morphological criteria, and demonstrating loss of E-cadherin by immunohistochemistry can be useful, but is not necessary or mandatory. We know, however, that there is some disagreement between pathologists already being described in the late 90s, and also very recently in Central Pathology Review of large clinical trials. Some work has been done already demonstrating that the use of E-cadherin immunohistochemistry does improve agreement between pathologists. Here left, you can see the work of Matthias Krisgen demonstrating improved agreement between pathologists and also agreement to a reference standard. And more recently, the work of Gabor Czerny illustrated that the use of E-cadherin led to a reduction of uncertain typing of breast cancer cases. So we were actually wondering what is still the problem then in current pathological practice. So we performed a survey recently asking pathology labs worldwide about their preferred practices. And so we first observed quite some heterogeneity and indications for performing immunohistochemistry, next to which combinations or antibodies they would use for an ILC. And then when we dig further into E-cadherin antibodies, we can saw that there were 11 different clones reported. Here you can see the 4 most frequent ones. And here for this clone, NCH38, we could see that up to 7 different concentrations were reported being used for this clone. Now, is this important? Well, here in this figure, you can see a schematic overview of the E-cadherin protein. And here the epitopes or the specific sites where all of these antibodies bind to. Now, this gets interesting because here you can see an overview of the CDH1 gene encoding for E-cadherin. And all these lollipops present reported mutations in this gene. So we have quite a variety of genes ranging from the extracellular part going to cytoplasmic. And also these mutations can have differences in the effect they have on the transcript. Now, as already highlighted, the interpretation of cadherin immunohistochemistry is not dichotomous, not black and white, but there is an entire spectrum in between of aberrant patterns that can be a source of misinterpretation and maybe even misdiagnosis. So this led to the question of our study whether differences in E-cadherin antibody clones in combinations with different types of mutations in CDH1 would lead to differences in staining patterns and possibly reporting and diagnosis. And so this was the setup of our Cadillac study. And so in this study, we collected a variety of E-cadherin antibody clones, also P-cadherin, beta-catenin, and P120-catenin. And first of all, our colleagues from the Netherlands, from Utrecht, performed specificity analysis of these E-cadherin antibody clones. So they performed a Western blot. And what you can see here in these Western blots is that you have a very clear, sharp line for the clone in yellow and in green, the ECH6 and NCH39, meaning that they have quite a good specificity, while the other three clones, you see that there are multiple bands at several Daltons, indicating that they might recognize some other proteins as well. And what was interesting here is that for clone 36, we could see suddenly recognition of a protein when E-cadherin is not present in these cell lines. And this was then identified as P-cadherin. So actually, clone 36 also cross-reacts with P-cadherin, even in the absence of E-cadherin. Then we collected a series of 49 cases of breast tumors with known mutation in CDH1. And so we've collected mutations in the cytoplasmic coding end, as well as the extracellular end, and also with various effects on the transcript. We collected them from the WSG ADAPT trial, as well as from the University Hospitals of Leuven. And then we performed immunohistochemistry for all of these clones on all of these cases. We held a central pathology scoring session in Leuven for 4 days, quite an intense duration. And we asked 5 experienced breast pathologists, as you can see here, to evaluate these cases. First of all, the pathologists had to evaluate these cases only on H&E. They had to make a diagnosis call. Was it a lobular breast cancer, NST, or mixed? And if there was an ILC component, they had to make a subtype call. In the next phase, the observers were provided with immunohistochemistry of all the E-cadherin clones, as well as the catenin antibody clones. They had to observe the staining patterns of these antibody clones, and then finally had to go back to the diagnosis and indicate whether the diagnosis was changed or not. And so the first thing that we could observe is that indeed, when we look at inter-observer agreement, that it's increased after immunohistochemistry. We then evaluated the inter-observer agreement between staining patterns of the antibody clones. And so we could see that for each clone, the inter-observer agreement was quite okay if you're looking at a complete membranous staining, so what you expect for normal epithelial cell or an NST tumor, and any absent staining, so which is considered abnormal for normal cells. What we also could see is that it was quite high, the highest for this clone, NCLLE-CAD. And no, actually what we see is that it was influenced by a suboptimal internal control. So the staining protocol that we got, a recommended staining protocol, was actually not performance efficient. And actually all the cases were almost always way too weak for what it was expected. So we'll not consider this further in my presentation, this code. Next, our bioinformatician, Francois Richard, tried to evaluate whether there's an association between the type of mutation and the staining pattern of E-cadherin antibody staining. First of all, we looked at the location of the defect. So here we looked at whether an extracellular defect versus cytoplasmic mutation would have an effect on the staining pattern. But here we could not see any significant associations between the absence of E-cadherin staining or a complete membranous E-cadherin staining. What we did saw, however, was that the allelic frequency, the variant allelic frequency of the mutation of CDH1 was associated with absence of E-cadherin immunohistochemistry staining. What we also observed were some rare cases, some peculiar cases, which I will highlight now. For instance, we have here case 35, which on morphology had quite a typical appearance of a lobular carcinoma, but then when performing E-cadherin and the catenins, we saw preserved proteins of the cadherin-catenin complex. But so this mutation actually did not result in the loss of the E-cadherin protein, but resulted in the loss of function of the extracellular binding site. So this protein could not interact with the E-cadherin protein of another cell, but it was still present. Case number 2 here with the red box looked NST under the microscope and also displayed preserved components of the cadherin-catenin complex by immunohistochemistry, but did have a CDH1 mutation. Now, in the end, the consensus diagnosis for this was still an NST case. And the reason for this is because the mutation here was situated all the way at the end of the cytoplasmic end of CDH1, not affecting the binding site of beta-catenin or P120-catenin. So the function of the entire complex was not compromised. The last case that I will highlight here is case number 26. And this is actually an example of what is called an ILC with tubular elements. So we have a tumor with loose and infiltrative cells next to some nests and tubule formations. And these tubules and nests, they stain for E-cadherin clone 36, but not for the other antibody clones. And then when we look to the p-cadherin, we could see that this is actually due to the cross-reactivity that we observed earlier on. Based on our observations, we try to come up with a recommendation to perform and to interpret immunohistochemistry. So this is a bit small for you, I guess, on the screen, so I've made a summary. So it actually all starts with morphological evaluation of the tumor at first. And we listed some histopathological criteria that should raise concern for ILC. Now, E-cadherin immunohistochemistry should only be performed if one of these criteria are present, but if we do not have all the criteria for a classical ILC. So in other words, if you have a pure textbook case classical ILC, we do not recommend to perform immunohistochemistry. When immunohistochemistry for E-cadherin has been performed, we always advise to evaluate internal control, which is actually a general principle in pathology and is actually very important, as we have seen for clone NCL ECAD. Then we evaluate the staining pattern in the tumor, and when it's completely absent, the case of ILC is made. So if morphologically you have suspicion of ILC, E-cadherin is concordant, then it's clear it's an ILC. But if your morphology is suspicious for ILC and you have a pattern that is not concordant with ILC, we would advise you to perform ancillary immunohistochemistry. And in this immunohistochemistry, we recommend to do it by means of beta-catenin or P120-catenin. And this is actually up to the up to the practicing pathologist to choose which clone to use, which one he or she is more familiar with. And so to conclude my presentation, we have confirmed, as has been demonstrated in literature, that immunohistochemistry can improve the diagnosis of ILC. We have illustrated that for some E-cadherin antibody clones, there is a reduced specificity for E-cadherin and cross-reactivity can be observed. We have observed that allelic frequency is actually one of the only determinants that is associated with an absence staining pattern and not necessarily the location of the mutation. And we have put forward recommendations for integrating morphological features and immunohistochemistry for the diagnosis of ILC, hoping to improve and standardize the diagnosis of ILC. And with this, I would like to thank all of the pathologists and the clinicians, bioinformaticians, and lab managers who participated to this study, and also the funding partners that made this work possible. I thank you for your attention, and I'll be happy to take questions if possible.
Great talk, Dr. Shepard. We can have a few questions for Dr. Shepard if someone has any. Maybe I can ask a question. Dr. Shepard, if you notice a single cell infiltrative growth pattern and you do E-cadherin staining and you have a very strong membranous E-cadherin staining, how do you deal with those cases? Do you consider that as aberrant? Or obviously, do you do P120 beta-catenin on those? And how do you interpret as the final diagnosis on such cases?
Yes, that's thank you for your question. That's a very good question. So if in the first place, morphological point of view, it's a clear-cut case of ILC, then we would not perform E-cadherin immunohistochemistry. But if indeed E-cadherin has been performed and you have a clear-cut, complete and strong membranous staining, we would advise you to perform additional immunohistochemistry by means of P120 or beta-catenin to assess the adherence junction.
The reason I ask this question is because we have seen these cases in our clinical practice, and we recently published on these. And we like to call them as lobular-like invasive mammary carcinomas, because if you do P120 and beta-catenin on these cases, they generally also show membranous reactivity, and they generally do not have E-cadherin or CDH1 mutations. Some of them do have CDH1 promoter methylation. So I was just wondering if you have come across such cases and what you do.
Yes, I read your publication, indeed. So here in our study, we've focused mainly on cases with a known CDH1 mutation, so not with other types of aberrations. So this was actually the aim of the study, to characterize staining patterns according to the CDH1 mutations. Now, in the case of the promoter methylation of the CDH1 genes, these are indeed other mechanisms that can give a preserved staining, a complete membranous staining of the E-cadherin and the catenins. Here we've only observed one case like that, with the exotic missense mutation in the extracellular part. Now here we've still called this an ILC. And this would be, I think, similar as into your paper that you referred to as a lobular-like invasive mammary carcinoma. But we would refer it to as a lobular carcinoma indeed.
Our next speaker is Dr. Terry King. Dr. King is Ann Dyson Professor of Surgery at Harvard Medical School, the Chief of the Division of Breast Surgery, and the Vice Chair of Multidisciplinary Oncology in the Department of Surgery at Brigham and Women's Hospital, and Chief of Breast Surgery at Dana-Farber Brigham Cancer Center. She's also the Director of Breast Cancer Personalized Risk Assessment, Education and Prevention Program at Brigham and Women's Hospital. Welcome, Dr. King. We are all excited to hear about your talk on LCIS.
Thank you. Thank you for the nice introduction, and thank you to Steffi for the invitation to be here today. She actually invited me to speak at the very first symposium in 2016, and unfortunately, 2 months prior to the meeting, I had a fall in my home, and I had a broken shoulder and a broken wrist, so I wasn't able to travel, but I'm happy to be recovered and to be back with you today. Great. So we're going to talk about lobular carcinoma in situ. Okay, push that one. Okay, apologies for that. Okay, here are my disclosures. Okay, so LCIS is near and dear to my heart. We both started our journey at Memorial Hospital, although LCIS is a little older than I am. So I'll take you back a little bit further than Dr. Schnitt. So the first photomicrograph of what we now call LCIS was actually published in 1919 by James Ewing, who was the director of Memorial Hospital at the time, and he simply described it as a rare mammary lesion of unclear significance. Fast forward then to 1941, and Dr. Schnitt told us that it was actually Foote and Stewart that actually coined the term lobular carcinoma in situ. And at the time, again, seeing this in mastectomy specimens of women who had infiltrating lobular carcinoma, they hypothesized that this was a precursor to infiltrating lobular carcinoma and recommended that women undergo mastectomy. And it wasn't until 40 years later that we actually started to understand something about the natural history. We saw that rates of ipsilateral cancer in women with LCIS who were not treated were lower than expected at approximately 1% per year. And these rates were not that different than rates of contralateral breast cancer. And in addition, about 50% of the cancers that were developing in these women were of the ductal phenotype and 50% were of the lobular phenotype. And so the combination of this data actually led to a movement then to consider LCIS as a marker of risk and not necessarily a precursor lesion requiring treatment. And so as this epidemiological data evolved, our clinical management also evolved. And so through the 1990s and into the early 2010s, we saw far fewer women undergoing prophylactic mastectomy and more women being comfortable with high-risk surveillance as we became more comfortable with actually talking to women about LCIS as a risk factor. And these are data from SEER illustrating the change in clinical management. We see a decline in mastectomy throughout the 80s and in the 90s, and an increase in the use of surgical excision alone following a diagnosis of LCIS. In the 1990s is when core biopsies were introduced. And again, we now know that most of the time surgical excision is performed simply to rule out an associated carcinoma. Now, we've also learned a lot since the time of Foote and Stewart with respect to these lobular lesions. We recognize that they represent a spectrum of change, ranging from the low end, where we have atypical lobular hyperplasia, to now the high end, where we have pleomorphic lobular carcinoma in situ. And we know that the differences between these lesions are both qualitative and quantitative. So if we look at ALH and classic LCIS, the cells themselves actually look quite the same, but if the cells fill less than 50% of the acinar units and do not distend them, our pathologists classify them as ALH. And when those cells fill and distend at least half of the acinar units, that's when it's called classic LCIS. And we don't actually start to see a change in the cells themselves until we get up into the pleomorphic grouping, where the cells are now larger, there's more dishesion, pleomorphism, and prominent nucleoli. And as we also learned from Dr. Schnitt, whenever things fall along a spectrum, there is a challenge with inter-observer variability. So this is just one example, 610 specimens that were sent for formal consultation to an academic medical center in Brazil. And we see here that most of the time, our pathologists do agree when the TDLUs are normal. There was 96% agreement. When they involved ALH, there was only 47% agreement. Classic LCIS, 69% agreement. And pleomorphic LCIS, although very small numbers here, only 20% agreement. Whoops. Very sensitive now. So this variability leads to some challenges for us in the clinic because our natural history data tells us that women with ALH actually have a lower risk of developing breast cancer than women with classic LCIS. So the classic teaching is that if you have ALH, you have an increased relative risk over the population of 4 to 5-fold. And if you have classic LCIS, you have an increased relative risk of 8 to 10-fold. Now, we don't have much data on the natural history for pleomorphic LCIS, but there's a lot of speculation that the risk must be higher with these lesions because they look ugly and they share some common features with DCIS. But we've had trouble really collecting these lesions to be able to study them to understand the natural history for two reasons. One, they are rare. So variants of LCIS only account for approximately 2 to 5% of all LCIS. And over the last 2 decades, our pathologists have had trouble really describing these lesions. They've seen lesions now that don't have all the classic criteria of classic LCIS. They don't have all the criteria of DCIS. And so they've been describing them with these various different names, which has made it really hard to collect samples for study. Now, thankfully, with the most recent version of the WHO, expert consensus came together and decided that really going forward, that we really should be having 3 categories. That is classic LCIS and only 2 histologic variants. That is pleomorphic LCIS and florid LCIS, with the hopes, again, that more standardization will allow us to learn more about these lesions. Now, today, I'm really only going to focus mostly on classic LCIS with a few comments about pleomorphic, but I do highlight here 2 really nice references if you want to learn more about florid LCIS. So what do we know? Well, classic LCIS is typically an incidental finding. That means, for example, a woman has a biopsy for mammographic calcifications. The calcifications are actually associated with sclerosing adenosis, but in the surrounding breast tissue, the pathologist identifies classic LCIS. We know it's frequently multifocal and bilateral, and the molecular alterations are quite well defined with loss of 16q. As you heard earlier, that's where the CDH1 gene lives, and so these lesions are E-cadherin negative. They've lost that functional protein. Natural history, again, we counsel women that it's a risk factor, but another term for that is really a non-obligate precursor. Not all women with LCIS will get breast cancer, but some will. And we know the risk is bilateral, again, both breasts, and the risk is equal for ductal and lobular lesions. And our challenges with classic LCIS is really, why do some women develop LCIS and others don't? And can we individualize risk prediction? With PLCIS, this is usually not an incidental finding. There is a mammographic abnormality or a mass that actually is the PLCIS. About 80 to 90% of the time, it's mixed with classic LCIS. And importantly, most patients with PLCIS already have breast cancer. And this is predominantly the lobular phenotype. We see many of the same molecular alterations as in classic LCIS. They are also E-cadherin negative. They can be ER positive or ER negative, although mostly ER positive, and they have some additional copy number gains and losses. They can be HER2 positive, and they can also be P53 positive, which we really don't see in classic LCIS. And again, our challenges here are that they are rare, and there's been a lack of agreement on the diagnosis, and so we don't know much about the natural history. So myself and others have spent a lot of time trying to characterize the risk associated with LCIS. As was noted earlier by Dr. Mukhar, you go to the clinic, you identify a problem, and you want to go back to your office or your lab or your team and you want to try to address that problem. So for the last 20 years, I've been trying to figure out why some women with LCIS get breast cancer and others don't. And so this is a large series that we put together when I was at Memorial, just over 1,000 patients who were undergoing surveillance over a course of about 3 decades. And you can see here that the rate of cancer development in this cohort is very steady at about 2% per year. Now, again, we wanted to see, well, can we predict who was going to get cancer? So we did a very detailed slide review, a nested case control cohort study, 72 cases, meaning 72 women that developed cancer versus 274 controls. We went back and looked at their original LCIS slides, and we were not able to identify any predictors of risk. Similarly, on multivariate analysis, we looked at all of the traditional clinical risk factors for breast cancer, and we could not identify anything that would separate these women into high and low risk categories. We did get a hint, however, that the volume of LCIS in the specimens may be important. When we classified volume as the ratio of the number of slides with LCIS over the number of slides reviewed, we did see an increasing hazard with a higher ratio of LCIS, but this has not been reproduced in any other data set that I'm aware of. So again, going back to the SEER data, we see from SEER that 10-year cumulative incidence of cancer after LCIS is about 11%, 20-year cumulative incidence, 20%. So this is very consistent with the original observations of about 1% per year back in the days of Rosen and Hagensen. Our rates in the Memorial cohort are probably a little bit higher because of that central pathology review confirming LCIS in all cases. We also see from SEER that about 20% of the diagnoses are DCIS and about 45% are stage 1 invasive breast cancer, and most of the invasive breast cancers are low grade, grade 1 or 2. We do again see that women with LCIS are equally likely to get a ductal versus a lobular cancer, but lobular cancers are certainly overrepresented in women with LCIS when we can think about the general population. So in the general population, lobular cancers represent only about 10% of all breast cancers, but in women with LCIS, they represent, again, about 30 to 40%. We saw this also again in the Memorial cohort, invasive disease, more common than DCIS, 12% versus 6% at 10 years, equal number of ductal versus lobular cancers. And importantly, the cancers that develop in women with LCIS are overwhelmingly ER and PR positive, which has important implications for chemoprevention in this cohort, which we'll talk about later. When we look at ipsilateral cancer versus contralateral cancer rates, we see that ipsilateral cancer rates are a little bit more higher, both in the Memorial cohort and in the SEER data, and that cancers that develop in the ipsilateral breast are more likely to be lobular cancers. Now, my colleagues at Memorial, they continued on this work after I left. And so with a larger population now and a little bit greater follow-up time, they were able to see an association between breast density and the risk of cancer in women with LCIS, such that women who have dense breasts have an increased hazard of 2.4 for the development of breast cancer. Also here again, showing the significant benefit of chemoprevention in this cohort, a 50% reduction in the risk of subsequent breast cancer with chemoprevention use. Again, there was no association with BMI, family history, menopause status, and other factors that we had looked at previously. Now, in addition to the natural history data in the clinic, we have risk models that we try to use to help women understand their risk of breast cancer. Two of these models, the Tyrer-Cuzick model and the BCSC risk calculator, do have a variable for the presence of LCIS. And so we decided to compare the output of these models to what we know again from the natural history in our large cohort. So here I'm showing you the calibration plots for Tyrer-Cuzick version 7 on your left and the BCSC risk calculator on your right. And we see that for Tyrer-Cuzick version 7, there's actually very poor calibration in this LCIS population with a very high overestimation of risk. And when we look at the discrimination, which is the individual patient level, the C index is only 0.49, so basically no better than flipping a coin. When we look at the BCSC risk calculator, we see better calibration. So most of those Kaplan-Meier estimate lines do cross the line of equivalency in the middle there. So modest calibration in the population. But again, when we look at this discrimination ability, again, one patient versus another, the AUC is only 0.59 at 5 years and 0.58 at 10 years. So just slightly better than flipping a coin. Now, we know that these models are imperfect. These models also are very poor in non-white women. And this is a very interesting analysis from SEER, where there is some evidence that actually Black women may have a higher risk of developing cancer after diagnosis of LCIS. Just under 19,000 patients here. 10-year incidence of invasive breast cancer ranged from about 8% in Asian patients up to 11% in Black patients, which was significant. And on multivariate analysis, we see that Black women are more likely to develop any subsequent invasive breast cancer with the hazard of 1.33, equally true for ipsilateral cancers or contralateral cancers. And interestingly, this increased risk was true for both subsequent ER positive and ER negative disease. So certainly more to be learned from this analysis. So again, myself and others, we've been trying to figure out, well, can we use some molecular markers or genomics to try to identify which women with LCIS are really the ones at high risk of developing cancer? And so this was some of the early work that we did when I was at Memorial. We performed CGH arrays to look at copy number variation in women who had LCIS and either synchronous invasive lobular cancers or ductal cancers. And this is just one example where you can see just with the eye on this copy number profile, the reds are copy number gains, the blue lines are copy number losses. And when you look at the LCS versus the invasive lobular cancer, it looks very similar. But when you apply a very sophisticated bioinformatics algorithm to these profiles, you can actually come up with a odds in favor or against of clonality. And clonality means that the LCS and the invasive lobular cancer arose from the same precursor cell, so they are related. So in this first series, we had 17 pairs, and in 40%, the algorithm predicted that the pairs were definitely clonally related, and about 30% they were equivocal, and about 30% they were independent. So we went on and performed a larger study then where we looked at copy number profiles as well as mutational profiles from many pairs of lesions. We had LCS and ILC, LCS and IDC, pairs of LCIS from the same breast, and pairs of LCS and DCIS from the same breast. So after DNA quality control, you can see the number of pairs that we were able to successfully analyze across those cohorts, representing individual cases in the second row. And at the end of the day, what our analysis suggested was that about 74% of paired LCIS and ILC were actually clonally related, and also similarly true for that multifocal LCIS. And interestingly enough, although a small number of cases, the algorithm predicted that 5 of 8, or 63% of LCIS and DCIS from the same breast were in fact clonally related, suggesting the possibility of a single precursor cell that then could progress down either the lobular or the ductal pathway based on the acquisition of additional genomic changes. And we also looked at mutational profiles. Again, we microdissected LCIS and ILC from women that had synchronous lesions and did targeted parallel sequencing. Here we see, again, not surprisingly, that CDH1 mutations are the most common mutation in LCIS and ILC. And PIK3CA mutations were the second most common identified mutation. Again, the rest of the mutations listed, or rest of the genes, listed along there with a lower frequency of mutations. And this is very consistent with what was reported from the TCGA. The European group that are running the Glacier study, which is a very large effort to study LCIS in Europe, they also reported that PIK3CA mutations were quite common in about 20% of their samples, and concluded that while these mutations seem to be an early event in lobular tumorigenesis, they do not appear to be a critical event leading to invasive disease. And in their work, they did have some suggestion that perhaps cyclin D1 overexpression was associated with risk. But again, to my knowledge, this has not been confirmed in another data set. So coming back then to, we have a lot of data, again, about classic LCIS, trying to understand risk. We have very little data, again, for pleomorphic LCIS. This is actually all of the data that we have on the natural history of this lesion. 132 cases reported in the literature. These are all small retrospective series. You can see the authors collected these cases over long periods of time. There was not standard treatment regimens, and there are different lengths of follow-up. But when you put them all together, we have 7 out of 132, or 5%, that have developed recurrent PLCIS, 10 or 7.5% that have been diagnosed with an ipsilateral cancer diagnosis, and again, various follow-up times. So this is really all we know about the natural history of PLCIS. There we go. Okay, so in the last few minutes, let's talk about some of the standard surgical or some of the standard clinical issues. So surgical issues are, what do you do when you have LCIS on a core biopsy, and do margins matter? Of course, chemo prevention I've alluded to a couple times, and then how should we be screening women with LCIS? So we no longer routinely perform excision for a core biopsy diagnosis of classic ALH or classic LCIS. There are several series in literature now. These are 4 of the studies that really changed the paradigm. The bottom study was led by my colleague, Dr. Knocklis, and with central pathology review by Stuart Schnitt. But all of these studies show that if you have good radiographic pathologic correlation and you exclude other lesions that would independently necessitate an excision, that the rate of finding cancer is about 3%, and the cancers that are found are low-grade DCS or very small invasive ductal cancers, and therefore we do not routinely excise classic LCIS on a core biopsy. We also have follow-up data now that this is a safe approach. So these are data from our clinic in Boston, 127 patients with lobular neoplasia that were followed. 25 of them did undergo excision, 100 were observed. And at 3 years, the actuarial rate of cancer development was 4%. But importantly, none of the cancers developed at the site of the previous core biopsy. 2 cancers were in the ipsilateral breast, but in a different quadrant, and 3 were in the contralateral breast. We have similar safety data now out of Memorial Sloan Kettering. They reported on 170 cases that were excised compared to 142 that were observed. And the 5-year rates of cancer development were 8.9% with excision and 10% with observation. So it is clear that women with LCS are at higher risk, but excising the core biopsy diagnosis of LCS does not change that risk. Now, this is in contrast to the data that's available for pleomorphic lobular carcinoma in situ. When you see PLCIS or one of the variants on core biopsy, it should be excised. Again, this is a compilation of what's available in the literature, many, many small retrospective series. But when you add them up, 255 cases with an upgrade rate of 37%. So again, most women with PLCIS actually have breast cancer at the time of the diagnosis. What about margins? Well, we've known for a long time that classic LCIS at the margin is not important, and our pathologists don't report LCIS at the margin for us. But PLCIS at the margin, again, raises more questions. Again, very little data. This is a compilation of the studies that have reported on margin status and risk of recurrence in women treated with lumpectomy. Again, various different treatments. Some received endocrine therapy, some received radiation, some didn't. When you put them all together, it does seem as that patients with positive margins or those with close margins defined as less than 1 millimeter have higher rates of recurrence of about 30%, whereas patients who have negative margins have a very low rate of recurrence of about 4%. And my colleague again, Dr. Nachlis, recently published on the presence of non-classic LCIS in lumpectomy specimens for patients having breast-conserving surgery, wanted to demonstrate again that it is safe for women to have breast-conserving surgery, even if they have one of these variants, and there was no increased risk in local recurrence. And again, a recent publication out of the Memorial Group has shown the same thing. So in summary, for classic LCIS, for core biopsy, only selective excision for those cases that are felt to be discordant, and we don't worry about margins. For PLCIS, we still need to routinely excise the core biopsies. There is a substantial risk of finding cancer. And when it comes to margins, we should be assessing the margins and we should be making reasonable attempts to clear those margins. Chemoprevention, we learned in the NSABP P1 trial in the late 90s that taking medicine can reduce the risk of developing ER-positive breast cancer. Women with LCIS get ER-positive breast cancer, and so chemoprevention is quite effective. This is, again, the data from the Memorial cohort. Women who took chemoprevention, the 5-year rate of breast cancer was 3.3% compared to 11.6% in those who did not. And the relationship remains strong at 10 years. So chemoprevention works in the real world too, not just in clinical trials. This was also reported out of the Boston experience by Kevin Hughes and colleagues. They looked at all high-risk lesions, but these are the data for 568 women with LCIS, again showing substantially reduced rates of cancer at 5 and 10 years with the use of chemoprevention. What about MRI screening? This is quite controversial. It seems intuitive that if MRI is more sensitive, that we should find more cancers. Well, we looked at this in patients with LCIS, 776 patients. Again, this was when I was at Memorial. It was not randomized, so the choice of whether MRI was being used or not was at the discretion of the physician and the patient. So 60% of patients were having both mammogram and MRI, 40% were having mammogram only. Again, not randomized, and the screening intervals were different. So in order to perform the analysis, we did a landmark analysis to accommodate for these findings. And when we looked at 1 year after LCS diagnosis, 2 years after LCS diagnosis, or 3 years after LCS diagnosis, we could not find any evidence that MRI increased the cancer detection rate. We did this adjusting for family history and age, and also sensitivity analysis for the number of MRs received. And again, we could not identify a benefit to using MR in this population. We similarly looked at this recently, again, now in the Boston cohort. all patients with high-risk lesions here, just about 700 patients, about a third of them had LCIS. Again, not randomized, 77% were having mammogram alone, 23% were having mammogram plus MRI based on the physician and the patient choice. Again, the groups are not matched, so we did propensity scores to match these groups. And when you look at the weighted 4-year breast cancer detection rate, was no different between MRI use and mammogram use, just at 3.6% in both groups. But there were significantly more breast biopsies performed during the surveillance period in the MRI group. And again, we saw this in the Memorial data. So MRI is a good test, but it's not a perfect test. So I think as we move forward, we really need to be thinking about how to make screening recommendations based on the biology of the disease that we're studying and not on a numerical risk. So in conclusion, for LCIS, so clinical risk prediction remains limited, but most women with classic LCIS will not develop invasive breast cancer, and there is a clear role for chemoprevention. We know that there are genomic similarities between LCS and ILC, but as yet, there's no clear marker to identify which LCS is most likely to progress. We have the recognition of these variants. We lack natural history data, and we should make pragmatic efforts for clear margins in this group. I didn't show any data, but there's no data for radiation use in this group, and I really like to try to avoid treating this like DCIS. And we need to continue to refine our clinical management based on evidence. Again, we've learned that we don't need routine excision for classic LCIS. We should really consider the risk-benefit ratio for MR screening and encourage chemoprevention. Thank you very much.
Great talk, Dr. King. I don't think we will have time for any questions, but thank you so much. Our next speaker is Dr. Luns Giraudy from the famed Institut Curie in France. Dr. Giraudy will be discussing how E-cadherin inactivation shapes tumor microenvironment. Dr. Giraudy.
Good afternoon, everyone. Thank you for giving me the opportunity to present my PhD work today. So, as you know, ILC tumor microenvironment has some specificities, including TIL, which are associated with a poor prognosis in this cancer. Another distinctive feature of ILC is the minimal stromal reaction that we frequently observe in this cancer. One of the main components of the stroma of cancer are cancer-associated fibroblasts. Our team has described 4 main CAF population in cancer, and especially in breast cancers, named CAFS1 to CAFS4. CAFS1 is the most abundant population, which is a myofibroblastic population only found in cancer, contrary to CAFS2 and CAFS3, which are also found in healthy tissue. CAFS1 express at high level FAP, and in fact, our team has demonstrated that CAFS1 are themselves heterogeneous at the single cell level. We can distinguish inflammatory ICAF, notably including detox ICAF, and myofibroblastic MyCAF, which include the most predominant type, ECM MyCAF, which express SDC1. At the phenotypic level, ICAF are FAP positive, anthraxR1 negative, and MyCAF are FAP positive, anthraxR1 positive. We have recently shown that there is a trajectory of differentiation from detox ICAF to ECM MyCAF, and this highlights the huge plasticity of this CAF S1 subpopulation. All of these findings have been established from analysis of invasive ductal carcinoma samples, and so far little was known for ILC. In this context, I started my PhD project with the aim to characterize CAF in ILC. And first, we have done immunohistochemical analysis, and to do so, we have established TMA for more than 150 ILC and 77 ER-positive invasive ductal carcinoma. The ILC was actually taken from a large retrospective series of 251 cases with RNA sequencing data available. We have done several IHC of CAF markers, including smooth muscle actin, which is a myofibroblastic marker, FAP, CAFS1 marker, Anthraxer1, a MyCAF marker, and SDC1, an ECM MyCAF marker. Regarding SMA, we can see that in ILC, there is a lower expression of SMA compared to ER-positive invasive ductal carcinoma, so the myofibroblastic content is lower in ILC. We wanted to know more about the CAFS1 cluster composition, and to do so, we have combined the analysis of several CAFS1 markers, and we have found that there is a clear enrichment in ICAF in ILC, and also there is a lower amount of MyCAF, including especially ECM MyCAF in ILC compared to ER-positive IDC. We have confirmed this result at the single cell level. Indeed, we have done 6 single cell RNA sequencing analysis of classical ILC, and we have compared of data with public data set of ER-positive IDC. And we have confirmed that there is a clear enrichment in ICAF clusters in ILC, and there is a lower amount of MyCAF cluster, especially a lower amount of ECM MyCAF. We wanted to investigate the CAFS1 populations in the whole series of 251 cases with the same single cell level resolution. To do so, we have used a deconvolution method called bias prism, which can deconvolute the precise cell composition from bulk RNA sequencing data and based on the cluster identified in our single cell RNA sequencing analysis. By this way, we have found that the composition of ICAFS1 cluster is highly heterogeneous across ILC cases. Very interestingly, the heterogeneity of the CAFS1 cluster in ILC is correlated with the histopathological heterogeneity of ILC. Here you can see clustering analysis based on deconvolution data of CAFS1 population, and you can see that there is some group of ILC that are enriched for some histopathological feature, including the histopathological type and the tumor cellularity. Indeed, the ILC of classical type with low cellularity have the lower amount of ECM MyCAF and the higher amount of detox ICAF compared to the other ILC. We have also compared the composition of CAFS1 with ER-positive IDC from the TCGJ using the same deconvolution method. And so we have found that overall the ILC have a lower amount of ECM MyCAF compared to IDC. All this result was validated at the IHC level. We next investigated if there is a mechanism that could explain the lower amount of MyCAF in ILC. To do so, we have performed in silico ligand receptor interaction analysis of our single cell data. And we have found that the cancer cells in IDC can interact via icadherin with the CAFS1 clusters, which express at high level integrin beta-1. In ILC, we do not see this kind of interaction. We wanted to test this heterotypic interaction between integrin beta-1 and icadherin with a functional co-culture experiment. So we have co-cultivated MCF7 and T47D cell lines with or without CDH1 inactivation. with ICAF derived from breast tumor samples. After 2 days of co-culture, we can see an increase of ECM ICAF reflecting the transition from detox ICAF toward ECM ICAF. Interestingly, the silencing of E-cadherin in cancer cells totally prevent this differentiation into ECM ICAF. We have the same kind of results with both cell lines, and also we have exactly the same results when we inactivate not E-cadherin in cancer cells, but when we inactivate integrin beta-1 in ICAF. So E-cadherin pathway is involved in CAF differentiation, and this can explain the observation that we have made on the tissue. We next wanted to explore the interaction between CAF and TIL, and we have found that there is that CAFS1 have an impact on the pattern of TIL organization. Indeed, the ILC with a margin predominant pattern of TIL organization are a higher expression of anthraxer1 marker and SDC1 marker by IHC analysis. And this pattern is also enriched in ECM MyCAF cluster of CAFS1 by deconvolution analysis. At the opposite, the infiltrating pattern of TIL was clearly enriched in detox ICAF cluster. This data was totally consistent with functional analysis, as we have shown that the ICAF was the unique population that can significantly attract CD8 T cell in a transwell immigration assay. On the clinical level, we have found that the infiltrating pattern of TIL correlated with a poor survival. So as TIL have not a good prognosis in ILC, we wanted to know if there is a mechanism that can prevent the action of TIL on ILC cancer cells. To investigate this, we have explored our single cell data, this time the lymphoid population, and we have found that in ILC, a population was lacking. This population is a CD8 T cell population that express integrin alpha E, which is a known receptor of E-cadherin. And in fact, this population corresponds to a previously described population, which is called T resident memory CD8 T cell. This population is known to be highly cytotoxic and an anti-tumoral population. This data was validated in larger cohort with deconvolution analysis. And very briefly, we have found by functional assays that both E-cadherin in cancer cells and integrin alpha in CD8 T cells are required to have an activation and cytotoxic response of CD8 T cell after a co-culture with breast cancer cells. So here I'll summarize my conclusion. CAF content in ILC is heterogeneous, depending notably on histopathological feature of ILC. Globally, ILC have more detox ICAF and less ECM ICAF than IDC. And this is linked to CDH1 inactivation in tumor cells, which limit the differentiation from detox ICAF to ECM ICAF. And this is due potentially via an heterotopic interaction between E-cadherin and integrin beta-1. We have found also that detox ICAF can attract TILs and correlated with a specific pattern of TIL infiltration. And finally, we have shown that the poor prognosis of TILs in ILC could be related to E-cadherin-related immune escape mechanism. And this mechanism could be linked to the lack of interaction between the tumor cells and T-resident memory CD8 T cells. So thank you very much.
I'm so sorry. We are running a few minutes late, so we won't have time for questions unless someone has a really burning question they want to ask very quickly. Otherwise, we'll move on. Yes.
Number of fibroblasts in a given tumor volume in IDC versus ILC.
The ECM MyCAF tend to be found in higher amount. Usually, when we detect ECM MyCAF, there is a lot of myofibroblasts, really activated myofibroblasts. So I think that there is a higher amount of fibroblasts in IDC.
Yeah.
But yeah, maybe we have to explore this in detail from our deconvolution database because it is not so easy to have this answer. Yeah, with IHE, it's not easy. Yeah.
Thank you, Dr. Girardi.
Super quick question.
Yes.
How does your data or your different CAF populations, does that segregate in any way with the overall subtypes that were ILC specific that were found by the genomics people? So hormone related, immune related, reactive.
So for the moment, we have not performed this analysis, but we have this molecular class in our data set, and so we will perform this analysis. Yeah. Thank you.
Very excited to introduce our next speaker, Dr. Osama Shah, who recently completed his PhD in Dr. Osterreich's lab right here in Pittsburgh. Dr. Shah will be sharing his work on mixed ductal and lobular carcinoma. Dr. Shah.
Good afternoon, everyone. My name is Samish Shah, and today I'll be sharing one of the projects that is related with invasive lobular carcinoma and that we have worked on in our lab. The manuscript for this is out in preprint, so if you want to know more details about the methodology, you're welcome to look at that. But with that, I will just start with the main topic, which is the spatial profiling of mixed invasive ductal lobular carcinoma. So as we heard from the previous talks, this is a special entity. It's in some ways, it's a variant of ILC. And in other ways, we think this might be a unique entity itself. So we heard a lot about the background on breast cancer, and we know that there are various histological subtypes that are summarized here. And then ILC and mixed invasive ductal lobular carcinoma account for a smaller percentage compared to the NSTs. And we also heard before that the invasive lobular carcinoma has distinct survival. Specifically, if you look at the long-term survival for ILC compared to NSTs, they do much worse, and they have more frequent late recurrences. ILC also looks different. We have talked a lot about this, but just a quick recap. If you look at the histology H&E slides on the right and left, we can see clearly ILCs are quite distinct. They're discohesive tumors growing in single file compared to the NSTs, which are more solid cohesive tumors growing in a bulky growth pattern. Importantly, now we know that ILC also has unique biology, and the most commonly enriched mutations are highlighted here: CDH1, PTEN, FOXA1, TBX3, and CDH1 is mutated in most of the ILCs. And this is a gene that makes E-cadherin. We again talked about E-cadherin before. It's an important regulator of cell-cell adhesion. So in summary, ILC has distinct biology, which has important clinical implications. First, the E-cadherin deficiency that we see in the invasive lobular carcinoma makes it hard to detect and hard to resect. And it goes to unique sites of metastasis as we learned before. It's a predominantly ER-positive luminal disease with low Ki-67, and it responds better to endocrine therapy and perhaps less responsive to chemotherapy. And the unique mutations that we see in ILC, they may be unique drivers of progression and resistance. Now we have this entity called mixed invasive ductal lobular carcinoma, which is defined as admix of NST with 10 to 90% of ILC component. So this is the latest definition from WHO. And within the same tumor, we have two histologic regions. We have a ductal region, which looks like NST, and a lobular region, which looks like ILC. Just like the pure subtypes, they also have E-cadherin differences, where the ductal is E-cadherin positive and the lobular is E-cadherin negative. So if you look at the PubMed reports, we can clearly see ILC and NST, when we compare mixed tumors with NST and ILC, they're very understudied. So if you look at the observed versus expected reports for NST and ILC based on their incidence, the ratio is 1. It shows that we see the same number of reports as we would expect for the disease of this incidence. But for MDLC, if you see the ratio, it's near 0. So there are hardly 43 reports on mixed tumors. And we would expect more. So it's quite understated. However, the key seminal studies that have come out, they have shown us some important findings. If you look at the genomic analysis of individual components, just a few studies that came out, they show that the individual regions, ductal lobular, can have distinct mutations. And then the lobular could have mutations in CDH1, which explains the loss of E-cadherin. The bulk molecular analysis has been limited, but the largest one from the TCGA study showed that MDLC are having some molecular features that are similar with NSTs, while some similarities with ILC. Just like the bulk analysis, the clinical analysis shows that they have, so there's a lot of contradiction, but the recent study from our group shows that clinically, MDLCs are more like ILC, so they're more ER positive, lower grade than NSTs. However, the in-depth analysis of the individual tumor regions is very limited. So this is where our study came in. We aim to do multi-omic profiling of mixed ductal lobular carcinomas. We took 3 approaches. For the first one, we used digital spatial profiling to look at the different regions separately. So as you imagine, this is a very heterogeneous disease. We had tried some other approaches, but this was, given that we have spatial technology, this is a really good approach to study a heterogeneous disease like mixed tumors. So we used E-cadherin as a marker to distinguish between ductal lobular regions, and we profiled each of them separately. So that was the RNA-seq. Second was microdissection of the ductal lobular regions for MSK impact panel DNA sequencing for mutation profiling. And finally, we had one case where we did single-cell RNA-seq. So you can see the cohort is quite small. That's one of the challenges that we realized, that identifying mixed tumors is quite hard. But this multi-omic profiling allowed us to answer some important questions in the field. Do the 2 regions have distinct transcriptome? Do they have different molecular alterations? And whether the tumor region shows single-cell heterogeneity. The 3 cases, very briefly, are highlighted here. They're clinical timelines. All of these were ER positive. The HNE and then E-cadherin P120 slides are shown on the right, highlighting the different regions that we identified, the ductal lobular regions, respectively. So this is the spatial RNA-seq data, and each of the 3 tumors has been separated, with the ductal lobular components also separated. And you can see, based on the top variable genes, we see mRNA differences, and the most striking differences shown by the MDLC3 ductal region on the very right. Importantly, we also looked at the molecular subtypes, and we saw there are differences between ductal lobular regions for the intrinsic molecular subtypes. And the most striking, again, was seen in the difference was seen in the MDLC3, where the lobular region was luminal A, while the basal was, the ductal region was basal. Importantly, for this particular case, MDLC3, the ductal region was the one that went on to metastasize to the brain and lung. So this is a quite interesting finding, that within the same tumor, we have 2 regions with distinct molecular subtypes, and it may pose different risk. In this case, the basal was, as we would expect, the more higher risk disease. We also performed differential gene expression analysis, and we looked at the pathways, especially oncogenic pathways that are distinct between the ductal lobular regions. And interestingly, we saw that estrogen-related pathways, estrogen response pathways, were enriched in the lobular, while the MYC-related pathways were enriched in the ductal. We confirmed this finding further with transcription factor analysis. And on the left and right, we have ductal and lobular regulatory networks, respectively. If you see in the subset below, which magnifies the key transcription factors enriched, we see ESR1, FOXA1, and FOXA2 related with estrogen, and then MYC and MEX related with MYC targets in the ductal and lobular pathways. Similar to our RNA analysis, we also looked at the mutations, and we saw that the ductal lobular components, especially in MDLC1 and 3, show very distinct mutations, clearly distinct mutations. In the case of MDLC2, we see some shared mutations, perhaps suggesting clonal relatedness, but there are still some mutations that were quite distinct. Importantly, as we would expect, the CDH1 was inactivated in all of these lobular regions. In the case of MDLC1 and 3, this was due to mutations, truncating mutations, while in the case of MDLC2, we found a promoter methylation. So this highlights perhaps another mechanism of inactivation of CDH1 in mixed tumors. The other important thing is many of these mutations that we identify, these are either prognostic or druggable biomarkers. So for the single-cell RNA-seq, there was a challenge that we realized. When we did single-cell RNA-seq on one of the tumors, we did not see any CDH1 differences. We were trying to separate the populations using CDH1, but we did not see that. Interestingly, in this particular case, MDLC1, what was the challenge was that the mutation that was resulting in an activation of the CDH1, it did not affect the mRNA, but resulted in loss of the protein. So that would make sense. We could not distinguish between the ductal lobular populations at the single-cell RNA-seq level. However, using the matched spatial RNA-seq data, we were able to derive signatures to deconvolve the populations into two ductal and lobular populations as shown here. And with further subclustering, we identified 3 robust clusters in both ductal and lobular populations, and the marker genes are shown on the right, highlighting that they had distinct biology. And given its single-cell RNA-seq data, we also want to look at heterogeneity of the pathways that we identified. So just like we before identified estrogen enrichment and MYC enrichment in lobular versus ductal in the spatial RNA-seq, we also saw the same thing in just one sample. So this was quite nice to see the same thing at the single-cell RNA-seq in this sample. But interestingly, when we look at the subclusters of lobular, we saw that there was just one subcluster of lobular population that had higher enrichment than the others. Next, we asked ourselves whether these subclusters may reside in a spatially distinct niche within the tumor. So given we have spatial data and we have single-cell data, we mapped these together using GSVA of the marker genes of the subclusters, and then mapped and they scored these spatial regions to map these subclusters onto these spatial regions. This perhaps shows us that indeed these populations may reside in distinct niches, and that's why they have different biology. The top marker genes are shown here for each of the subcluster. So in summary, we performed multi-omic profiling of a subset of mixed cases using digital spatial profiling, genomic profiling, and single-cell profiling, and looked at the differences between biological features of ductal and lobular tumor regions. We saw distinct transcriptome and distinct intrinsic molecular subtypes, especially in one of the cases where we had a basal and luminal A disease. We saw enrichment of estrogen response pathways in the lobular, while MYC in the ductal. And then we also saw distinct actionable mutations in the lobular and ductal regions, and CDH1 was inactivated in all the lobular regions. As you can imagine, this heterogeneity is quite interesting, and it also has clinical relevance for the subset of patients, and it can complicate the patient prognosis and result in therapy dilemma. How do we treat a patient which has 2 regions that look different and are either luminal in one of the cases and basal? Do we give them same treatment? Do we give them different treatment? And then finally, the conclusion from this study is that we should profile the individual tumor regions within the MDLC for more precise treatment. I'd like to thank my mentors, Adrian Lee and Steffi Ostreich, and our lab, and to all the patients and advocates who support our research and donate their tissues. With that, I will thank everyone and take any questions.
Great talk. We are supposed to have coffee break at 2:45, but we can certainly take a few questions.
That was a really beautiful talk. With a small sample size and early days, it may not be yet time to answer this question, but I was really struck by the definition of being an admixture with 10 to 90% of ILC. Like philosophically, do we think it matters? Does 10% do something different than 90%? Or is it binary? Is it there? Is it not?
No, I think that's a great question, because if you change this definition a little bit, more ILCs could be defined as mixed. And maybe some NSGs could also be defined as mixed. The WHO definition actually has changed. I think before it was maybe more than 50% of ILC component. So I think this is one of the reasons why we see a lot of contradiction between the clinical studies. Some say we have the MDLC are more similar to ILC. Some say it's more similar to NSTs, but it's not clear. So perhaps we need better criteria to define mixed, and then look in the subclasses.
Sure, Dr. Schnitt want to comment. 10 to 90% is correct. So for calling anything mixed, you need more than 10%. That's a WHO definition.
In light of the data you were showing, I was wondering why you call them mixed and not coexisting.
Yeah. So mixed is maybe in some ways it could be a misnomer. It could be a broad term that defines complex sets of diseases. We actually looked at a larger cohort, and Dr. Bhargava saw that a lot of complex subtypes or complex histologies of ILC, like pleomorphic, they were called as mixed. So it seems like there may be a group of diseases that are if we don't know what they are, they may be labeled as mixed. And then out of those large number of cases which we actually found as mixed, there were different types of mixed. So I think this is much more complex than.
Maybe one approach to answer to your question is that gross examination of the specimen, there is just one nodule. And so we are used to name it mixed, but we understand from biology that it can be coexisting clones or intermixed. But I think it's more simple for us to
Yes.
name this.
They may be collision, they may be intermixed.
So I think that's what maybe some people are wondering. The case 1 and case 3 you showed had vastly different
Mutational
expression in ductal and lobular. So do you have any idea what the collision type or more intermixed type of mixed?
Yeah. So based on the H&E, when we looked at this, all of them had spatially distinct ductal lobular regions with some intermixing. So initially we thought these are really interesting cases. We can look at each of these regions. Turns out after we did the genomic analysis, based on just the mutations, MDLC2 was the case which Dr. Bhargava is pointing to. Seems like that was the only truly clonid-related mixed tumor. So the other two seems like they're collision tumors, but we are doing more analysis at the copy number and also at the single nucleotide variations or SNPs to see whether there is some other similarity, maybe early shared copy number events or SNPs that could answer that question.
Oh, one more.
Sorry, just a quick question.
Yes.
So these ones you were classed as mixed ductal lobulars. And in Dr. Schnitt's talk, different pathologists were classing the same tumor as either tubulo-lobular or mixed ductal lobulars. So how are these different from tubulo-lobulars? How do you distinguish?
Yeah, Dr. Bhargava.
Dr. Schnitt, you want? So the classification used was based on E-cadherin and P120 staining and obviously the morphology. Some people don't even consider tubulo-lobular as a variant of lobular cancer because there are papers that have been published about tubulo-lobular having membranous E-cadherin and P120 expression. Something similar now is called ILC with tubular elements where you can see small tubules, but they are also E-cadherin negative. So yeah, the E-cadherin positive ones are now called, yes. There is E-cadherin to P-cadherin switching in ILC with tubules. So you see increased expression in those ILC with tubule cancer.
Okay, thank you.
Thank you.
All right. Good afternoon, everyone. We're going to get started on session number 2 this afternoon. My name is Becca Riggins. I'm an associate professor of oncology at Georgetown's Lombardi Comprehensive Cancer Center. And I'm really honored to co-chair or to chair this session this afternoon on E-cadherin and the ILC tumor microenvironment. These are all words and acronyms we've heard a lot today. And so I'm going to leave it to our 2 fabulous speakers, but I wanted to introduce sort of a different lens or framework to think about this. And so when I had a younger child and I went to her elementary school classroom and tried to explain what it is that I did for a living as a breast cancer researcher, I brought pictures. And I didn't do a very good job of explaining what those pictures were, but what was up on the walls of the classroom were different ecosystems. We had rainforests, we had deserts, we had oceans. And I realized, hey, wait a minute. These pictures, these tumors, lobular breast cancer and ductal or no special type, are really ecosystems, which are living things that interact with each other as well as their physical environment. And I picked this example because our 2 speakers in this session are going to highlight both aspects of those living things. We're going to start with Professor Dirksen from Utrecht, who's going to talk about how these living things, how these cells interact with each other, or rather, when we lose a key capacity to interact with each other, that molecule E-cadherin, and what happens within the cell when that is lost. Our second speaker this afternoon in this session is going to be Professor Val Brunton from University of Edinburgh. And so she's going to talk about how those living things interact with their physical environment. In this case, a particular type of cell in that environment called cancer-associated fibroblasts, which we've just heard a little bit more about earlier, but now we're going to get into the cell biology of this. And so with that, Professor Dirksen, the floor is yours.
So I'd like to thank the organizers again for the kind opportunity to speak about our research, what we're doing in Utrecht. Title speaks for itself. Buckle up because it's hardcore cell biology. I'll try to keep it as decent as I can, but you never know. So first things first, the most important slide, the people who did the work, Max Retze, who's a graduate student on the picture, and Lotte Ensring, who's a biotechnical engineer, did most of the work. And we got a lot of reagents from René Bernards, Carlos Caldas, Yoshimi Takai, and Noboru Ishiyama. So thanks to them. So I work on a Mac and I don't use PowerPoint. So we have to thank Bill Gates if this gets messed up completely. So this is the junction I conveniently already introduced this morning. So you have a big complex of an E-cadherin molecule, which is transmembrane, and it binds to all kinds of catenins. And we already know that if you lose E-cadherin, you develop ILC. So what happens? The first thing that happens is you get actomyosin contractions. So what I was telling this morning, the cell collapses because it cannot undergo cell-cell contacts anymore, and you get hypercontractility due to actomyosin contraction. And this confers a phenotype called anoicus resistance, which basically means that the cell can survive without attaching to anything else, which is something that some cancer cells do. Now, the second major thing that happens, this is a real pain. I don't know what to push anymore because the pad? Yeah. Okay, sorry. So the second major thing that happens is the relief of the repression on growth factor receptors. So what happens if E-cadherin gets lost? The repression on these growth factor receptors is gone, and they get really sensitive towards activation through growth factor receptors. That also activates a survival pathway, but it's more general. It's not just the anchorage independence that survival that is affected, but the general survival. And the third thing that is really important in this disease is transcriptional de-repression. So what happens, P120 gets into the cytosol and a nucleus once E-cadherin is knocked out, and it activates a transcriptional activation program through Kaizo. And we think this is the main player that activates ID2, and ID2 represses cell cycle activation through binding to hypophosphorylated RB, which we published last year. Another take-home message of these three phenotypes is that, and I really want to emphasize this, also given the other talks this morning, the activation of the growth factor receptors is independent of activation of PI3 kinase through mutation. So if you want to have patients eligible for trials whereby you inhibit AKT, you should not be basing this on a PI3 kinase mutational status, but looking at E-cadherin mutation because it renders this pathway active. The bottom line of all this is that you activate motility, invasion, metastasis, dormancy, and long latency relapses, the full package that belongs to ILC. Right, so we know that 90% of ILC show loss of E-cadherin expression, but there's just also 10% that do express E-cadherin, and there was a nice example of Stuart this morning as well. So we know a little bit what goes on. These are really due to rare mutations in E-cadherin. We have an example of this. So we got some samples from the ICR in London from Claire Isaac. It's a primary ILC sample, which you can see up here. It looks very ILC-ish, and it's also very E-cadherin positive. We were able to establish patient-derived organoids from this patient. And we found out there was a mutation in S180Y, so a transition from serine to tyrosine in 180. And the residue 180 is present in the extracellular domain 1, which confers the interaction between E-cadherin molecules. So together with Pere Roca Cusax, who is a collaborator from Spain in a mechanical control consortium, we modeled the impact of this mutation on the E-cadherin molecule, and that's what you can see here. So this is the wild type, this is the S180Y, and the tyrosine basically sticks out and it messes up the interface between an E-cadherin and an E-cadherin molecule. So you cannot get in-trans activation of the binding anymore. So this is why you end up with something that looks really lobular, but it's E-cadherin positive, and the whole molecule is there, but non-functional anymore. But these are really rare, and they don't explain the bulk of the disease, which is, and this is something I stole from a paper from Rice Philo, on average, you find about 60 to 70% mutation of E-cadherin in lobular breast cancer. So that still leaves, however you want to look at it, 20 to 30% that cannot be explained by E-cadherin mutations. So we wondered what is up, and does this give us insight on the biology of ILC and how this complex can be inactivated? So in other words, are there alternative driver genes that can provide the hit for ILC? So the first thing we looked at was the low-hanging fruit, which was alpha-catenin. Alpha-catenin is a molecule that doesn't bind to E-cadherin, but it binds to beta-catenin, and it provides linkage from E-cadherin to the actin cytoskeleton, so it can push and pull. Now, already a long time ago, this was described by Rachel Hazan in 1997 already. So she noticed that there were biallelic inactivating mutations in a cell line called MDA-MB468. And then later on, the Rotterdam group published 4 cell lines which had the same thing, including this 468. So biallelic inactivating mutations in alpha-catenin of cell lines of whom they thought were more lobular-like. So we went into the literature and we looked what kind of mutations are present within TCGA. And we picked up a couple, and we looked at the amino acid changes that would confer a stop or a frame shift, so the high hitters, and then looked at the mRNA expression levels, which are up here, and then picked the ones that are really low in mRNA. So the idea was you inactivate alpha-catenin through mutation, the mRNA is low, so there's no protein, so does this have an effect? The good thing about TCGA is that they provide a pathological histology. So you can look at the slides and see whether the pathology was actually paying attention during the initial diagnosis, and they weren't. So basically what you see here is a couple, 4, that are mutant, low in mRNA, and this is basically what you see. I mean, the upper one, yes, I can understand that someone would call this ductal or ductal-like, but the bottom one is clearly lobular. And they were stated as IDC NOS in TCGA. So these things are being misdiagnosed. So that's already a good sign if you have the perspective that alpha-catenin is important in this. So what we did was the functional experiment. We knocked it down in a cell line, both mouse and human. It doesn't really matter. So you see the knockout here. It's a knockdown, sorry. It's a doxycycline inducible knockdown of alpha-catenin. So alpha-catenin is gone. And what we then usually do is we put the protein back to see if we can restore function. That both gives you a good indication that you're actually looking at the right things protein-wise, and that the knockdown is specific. So that's what you see here. You can restore alpha-catenin. So the phenotype that occurs is, this is the control. All the cells are nicely compacted. What you see is E-cadherin in green and alpha-catenin in red. So the co-localization is yellow. So everything is as it should be. And if you then knock down alpha-catenin, you see that you end up with an E-cadherin positive phenotype of loose cells, which look, you cannot say ILC-ish because this is a cell line, but still you see that the cell-cell interactions are lost. And we can completely rescue that if you put back a non-targetable alpha-catenin construct. And now important for us is this actomyosin dependence. So we know that ILC is actomyosin dependent when it comes to anchorage independence. And what you see here is if you put these cells in anchorage independent conditions, both mouse and human origin, and you knock down alpha-catenin, they activate their anoikis resistance, which is this. And then if you inhibit actomyosin contraction either through Rho or ROC blebbistatin or actual F-actin depolymerization, you see that the anoikis resistance is lost. So basically, anoikis resistance is completely dependent on actomyosin contraction, which is very ILC-ish. So this is all in vitro. So we went to a mouse setup where we basically used conditional stem cells from a mouse that had a conditional p53 allele in them. We then transduced the cells with an inducible construct targeting alpha-catenin, put the cells back into a mouse, waited until the cells started growing, and then put the mice on doxycycline. So basically, you wait until the tumor starts forming, then you give the alpha-catenin knockdown signal, and you see whether this has an effect on tumor growth. And it did. So this is basically the control mouse. You see alpha-catenin in brown, so alpha-catenin is still there. You get these nice cauliflower type circumscribed growth patterns. And if you knock down alpha-catenin, you see that the cells are completely loose. And with a bit of imagination, you can see some trabecular formation or single file formation as well. This is not classical ILC by any means, but it's sort of an in-between ductulo-lobular phenotype, if I'm excused by the pathologist, if I can say such a thing. It looks very lobular, at least to a cell biologist. So what happens biochemically, you get clustering of an E-cadherin molecule that is not functional anymore. So it's on the membrane, but it cannot attach to anything anymore. So the cell collapses. So phenotypically, you get the same thing as an actual ILC. Okay. This is just alpha-catenin. This was just a lucky guess based on literature. So we wanted to do a sort of biased approach to see whether we can find new things. So we looked at the whole junction. So this is the adherence junction, which is alpha E-cadherin dependent. Next to it is the tight junction. We know that the tight junction is important for stabilization of the adherence junction. So this is something you should look at. And then there's the desmosome, which only forms if this whole complex is stable. So basically, this is epithelial adhesion. Based on literature and our personal preferences, we picked about 100 genes which we thought would be interesting and important for this mode of action. And then we asked the question, what if you take an ILC cohort and focus on the E-cadherin wild type ILCs and then look at additional mutations? Can you find something? And everything I'm going to show now is posted on bioarchives, not published yet. So we got the samples from René Bernards and Carlos Caldas, which were about 154 samples from the RADA cohort. They gave us the DNA. We made an exon capture of about 100 genes. In total, we had 146 eligible samples. About 84 were E-cadherin mutant, which is about 60% what you find in literature, so that's good. And then 62 were E-cadherin wild type, which is about 43%. And in this cohort, we found about 308 candidates, and we think of candidates of genes that play a role in either tight junction, adherence junction, or desmosomal formation. This is a spectrum of mutations we find, so you can look at your favorite thing. What we did, we looked at the low-hanging fruit, which is basically the stops and the frame shifts and the mutations that actually confer a negative phenotype, so loss of the protein. And today I will focus on one protein, one gene that we found, and that is ephedrine. So the patient was E-cadherin wild type and had an ephedrine mutation, and this resulted in a frame shift of the gene. Now, why ephedrine? Ephedrine has been implicated in a nectin-based junction, and nectin is a bit in between the tight junction and the adherence junction. And there were like two churches. One said you need a nectin-based junction in order to get an adherence junction, and the other said you need an adherence junction in order to get a nectin-based junction. I couldn't care less, but we had to look at it anyway because it does something with the effectin cytoskeleton. But what does it do with the effectin cytoskeleton? So we started this in 2017-18, I think. COVID hit, and then we got scooped by the Japanese, unfortunately. What they did is look at the exact same thing, not being interested in cancer at all, but what they did is they looked at this big protein, which is ephedrine, which has a C-terminal portion, which is really interesting because there it binds to nectin, to ZO1, which is a tight junction protein, to alpha-catenin, and to effectin. And that's the business end of the protein. So what the Japanese did, they found that it bound to alpha-catenin, and which is basically what we also found, but they beat us to it. Fortunately, this was the final figure of their paper. So they didn't understand what was going on, given all the question marks. So they found that it bound to alpha-catenin, but they didn't know whether it was pushing or pulling or whether it was absolutely required for epithelial function, let alone cancer. And so there was still a lot of work to do for us. So back to the mutation we found, it is a frame shift on position 6 to 9, which results in a stop at 630. So one could expect that this leads to loss of the entire protein. And in order to check that, we looked at the patient sample. So this is the ILC from the given patient I've been talking about. And this is the staining for an NST as a positive control. You see E-cadherin and ephedrine co-localizing on the membrane, which is what you would expect. And what we see in the patient is that ephedrine is indeed gone with the antibodies we have, but E-cadherin is still retained. So it looks like it's an E-cadherin positive ILC. You can see that the formation of the junction seems to be sort of intact, but there's also aberrant clustering, which gives the indication that the junction is not functional. But ephedrine is gone, so we assume that this was a loss of function tumor suppressive phenotype. So then we do the obvious experiment. We knocked it out in a cell line. We used MCF7 because reviewers like this one because it's ER positive. As a control, we took E-cadherin. E-cadherin is knocked out, which you can see here. And here we knocked out ephedrine, which is this band. It's the upper band. You have to trust me. I'll show that in a minute as well. So basically what you see, the wild type gives a nice clustered epithelial phenotype. If you knock out E-cadherin, this happens, you get ILC type cells, which you already knew as a positive control. And if you do an ephedrine knockout, you see almost a phenocopy of the E-cadherin phenotype. So the cells get loose, they cannot stick together anymore, and they don't form junctions. This results in anoikis resistance, just like ILC. So this is E-cadherin positive control. They are fully anoikis resistant. And if we do an ephedrine knockout, we get an in-between phenotype, but quite resilient to anoikis. So that's 0.8, which is quite high. And again, we looked at actomyosin contraction, and sure enough, the anoikis resistance is completely dependent on inhibition, on activation of Rho kinase and Rho itself. So indeed, it's an actomyosin contraction phenotype. So we still think we're on the track to explaining why this is an ILC. Now, what is going on? So you have a protein that is basically sticking on E-cadherin and doing something with the effect inside the skeleton. We looked at detail, and so this is the cell biology I'm going to show you. So these are cells in culture in which we stained for ephedrine and E-cadherin. And what we then do is we do perpendicular line scans with a confocal microscope across these borders and then quantify the signal in this region. And this looks like this. So in a control situation, you see perfect overlap of the E-cadherin signal in red and the F-actin, which is the cytoskeleton, in white. And then you can quantify the full width, half max density of these plots and see that there's co-localization. And what we saw in the ephedrine knockout is basically that the tension is gone. So the junction is still there, but the F-actin cannot build their forces anymore. So you get like a wobbly bit of F-actin next to it. And you can see that here as well. It's very wide, and that is quantified up here. So we did a lot of cells, and it looked like a big difference. So this is your control situation. And you see that the distance is widened if you knock out ephedrine. So it basically comprises a tension phenotype. So what we then did is to examine at the business end of the protein, where does the protein bind and what is important for binding to the effectin cytoskeleton. So in other words, this is the whole protein. And so what we did was mutation of either the binding domain to F-actin, or we removed the CC terminal end, which is the coil region that has the alpha-catenin binding site, or we removed the entire C terminus and just asked the question, what is important for binding and what is important for being lobular? And this is just a biochemical verification of the introduction of the protein. So mind you, we knocked it out, and then we put these truncated fragments back. So basically, this is what happens, what I just showed you. You get an acquisition of these loose cells, and this is what happens if you put back the different mutants. So with full length, you restore the entire phenotype. The delta Fab, to our surprise, which is the F-actin binding domain, does not have an effect. So you get a complete restoration of the epithelial phenotype, but only the coiled coil and the entire C terminus gives you the phenotype. So something in that region, and especially the coiled coil, which we think is the alpha-catenin binding region, is absolutely essential for its function in providing the junctional integrity. So we did the same trick with all these mutants, did all these line scans, and sure enough, you can see that in the delta CC and the delta C terminus, the effectin linkage is messed up, whereas in the control situation, you see that it's nicely one-on-one, and the delta Fab is also completely restoring the phenotype. So it's the CC portion that is the business end of the protein. And this is the quantification of this. So again, in red, it's this protein, it's missing only the CC terminus, and that's sufficient to drive the phenotype. So next we do, that's what we always do, we do mouse experiments. So basically we do MIND, which is something everyone does. It's been pioneered by Dan Medina, I think back in 2011. So you inject tumor cells directly intraductally. So this is a more physiological context, and then you wait until tumors grow out. And that's exactly what we did. We looked at growth. There's not much of a difference when you do either the knockout of ephedrine or the restoration with the delta CC or the delta C terminus. So the growth is about similar-ish, but there are big differences if you look at morphology. So this is the wild type. So these are MCF7 cells transplanted intraductally into a mouse and waited until tumors grow, and they look like this. So it's a circumscribed tumor, not very invasive. And this is the ephedrine knockout. So you already see you get these strand-like formations, and it looks more invasive. This is a blow-up of the HNE, and you can see these single file structures starting to invade. We also looked at the truncated versions of the CC and the C-term, and you see the exact same thing. So you see these ILC type growth patterns, and you see some invasion of these cells into the parenchyma, where you don't see this in the MCF7 controls. Now there's clear invasion. This is delta ephedrine. You see some tumor cells crawling into vessels here. So there's active metastases of these samples going on. And this resulted in a really big difference in lung metastases. So this is the wild type MCF7 control. If you put these mice on estradiol, they give some metastases, which is about this. And this is the quantification of that. But if you then either knock out ephedrine or the delta C terminal, you get the same phenotype. So the linkage to F-actin and to alpha-ketene is absolutely essential to give you this phenotype. And that results in more dead mice quicker when you look at the kill curve. So we can add this to the entire scheme. You have wild type, which is, we think, more ductal, and then you have a classical cadherin, which in which E-cadherin is lost. You have these rare mutations of alpha-ketene and cadherin that give you a lobular type phenotype, and we now add ephedrine to this scheme. And I do have to add that we're probably chipping away at the 10% that are E-cadherin positive. So we still don't know what's up with the 20% that have lost E-cadherin but are still a wild type. So what it boils down to in ILC, and this is, I think, the most important slide, from a biochemical point of view, we look at ILC to a classical type, which has nothing to do with morphology, mind you, from our perspective. So I think a classical type is a type that has E-cadherin mutation, and that leads to loss of E-cadherin. It's either a nonsense mutation or a frame shift or something epigenetic, which we don't understand. The important thing is you get F-actin disruption, growth factor receptor activation, and cell cycle repression. So that's the classical biochemical ILC for me. Now, the other, which we then call non-classical, is something that looks very much like ILC, but biochemically is probably not an ILC. So it has the F-actin disruption and you get the contraction, but it doesn't have the growth factor receptor activation or the cell cycle repression. So in other words, if you want to treat patients, I think in the near future with AKT drugs, and you have an E-cadherin positive ILC, it's probably not eligible for the drug because it's biochemically differently wired, more towards an IDC type. So it has clinical ramifications towards breast cancer diagnosis and treatment. Okay, I think with that I'll stop. If you really like this kind of research, I'm organizing a meeting next year. It's a Gordon Research Conference in the middle of nowhere, but always great fun. And please subscribe. Yeah. Okay, thank you.
Thank you, Patrick. That was lovely. I know we were running a little behind, but I wanted to give anyone the chance if they had a question. Yes.
Thank you, Patrick. It was really amazing.
Thank you.
Question from a pathologist. Do you have any antibody against Afadin available for diagnosis? I mean, you think that we should have it also? Test it?
Well, yes and no. There is an antibody, but the antibody is not up to standards.
Okay.
It's directed to the C terminus, so we might miss the N-terminal truncated version, which probably isn't there, but still. The antibody is really bad, so not good for pathology. And I think the penetrance of these types of mutations is so low that for regular diagnostics, it's more hobbyism for biochemists, maybe.
Got one more quick question.
Maybe I missed the information, but how many cases do you have mutated for Afadin in the Michaud series?
Well, in our series, we just found one patient with a mutation. If you look in TCGA, it's about 4%. So it's quite substantial, yeah.
Okay.
So there might be a group within the ductal cohort that are actually lobular and being completely misdiagnosed, yeah.
Thank you.
Thank you, Patrick.
So we move on to our next speaker, which is Ariella Henke. She's from UT Southwestern. She got a PhD working on ReBs and mTOR, and then went to Vanderbilt to work with Carlos Arteaga on HER2 mutations and clinical relevance. Today she's going to talk about therapeutic targeting of HER2 driver mutations in ILC.
All right. Thank you so much to the organizers for inviting me to present today. It is such an honor to be here at this really special symposium. So we've heard a little bit about HER2 so far. I think most of you know that you've heard about HER2 positive breast cancer, what we call HER2 positive. So what this really means is amplification of the HER2 gene, causing many copies of that gene, leading to overexpression and very high levels of the HER2 protein. Now, HER2 is a growth factor receptor. We've heard a little bit about growth factors and growth factor signaling. It is also a receptor tyrosine kinase. So I'll be talking a little bit about its kinase activity, which means that it can phosphorylate both itself and some other targets that activates a cascade of signaling molecules to promote cell cycle or cell proliferation. So just because a tumor is called HER2 negative does not mean that HER2 is not there. And today what I'll be talking about is an alternative way of activating HER2. So this is by a mutation in the DNA sequence of HER2. This usually occurs in the absence of HER2 gene amplification. And so HER2 positive breast cancers are primarily treated with anti-HER2 therapies such as Herceptin or trastuzumab and several others. And HER2 mutations are sensitive to some, but not all HER2 inhibitors. Now, in addition, HER2 amplification is almost exclusive to the HER2 positive clinical subtype of breast cancer, whereas HER2 mutations occur in all clinical subtypes of breast cancer. So for ER positive breast cancer, HER2 mutations are found in about 5 to 10% of ER positive metastatic breast cancers, but they're highly enriched in lobular breast cancers. So about 10 to 20% of ER positive lobular breast cancers have these activating mutations in HER2. And HER2 mutations both in ductal and lobular cancers promote resistance to endocrine therapies. Now, HER2 mutations can also occur in about 7 to 10% of metastatic HER2 amplified breast cancers. And here they're associated with resistance to anti-HER2 therapies such as trastuzumab and pertuzumab. HER2 mutations are pretty rare in triple negative breast cancers, but they're highly enriched in the luminal androgen receptor subtype, as well as in triple negative ILCs. And I think there's a lot of overlap between these two groups. So importantly, the only way to identify these HER2 mutations is by genomic tests. So these are mutations that are in the tumor. They're not hereditary. And the only way you can find them is by DNA sequencing, next-gen sequencing, genomic testing, whatever you want to call it. And to find them, you could either do sequencing on the tumor tissue or circulating tumor DNA. So I'd like to start with the case of a patient with ER-positive metastatic ILC progressing on standard therapies, including aromatase inhibitors. And next-gen sequencing found that her cancer had a HER2 L869R mutation, which back then, this was 2014, that mutation was not characterized. So in the lab, we showed that this mutation indeed activated HER2. It induced phosphorylation of some of the downstream targets of HER2, activating the PI3 kinase AKT pathway and MEK-ERK pathway, and also induced oncogenic growth, which was blocked by, not by one HER2 inhibitor, but it was blocked by another HER2 inhibitor called neratinib. And right around this time, the SUMMIT trial was opening. So this is a basket trial testing the HER2 inhibitor neratinib in HER2 mutant cancers. And this patient had a very nice response, first to single agent neratinib, followed by another response to the combination of neratinib and the estrogen receptor antagonist fulvestrant. So after about 14 months of nice response to this treatment on neratinib-based therapy, the tumor progressed. And at that time, circulating tumor DNA sequencing found, in addition to the original L869R mutation, a new mutation in HER2 had popped up. And this was a HER2 T798I mutation. And we immediately noticed that this mutation was homologous to other so-called gatekeeper mutations and other kinases, such as EGFR, T790M, and lung cancer. And we found by computational structural modeling that this mutation actually blocked the ability of the small molecule neratinib in green to bind to that binding pocket in HER2. But there was another HER2 inhibitor that was a little bit smaller than neratinib. So this was afatinib, and it was still able to fit in that smaller binding pocket with the mutation. And indeed, cells expressing the gatekeeper mutation were resistant to neratinib, but sensitive to afatinib. So then the patient was treated with a combination of afatinib and letrozole and had another durable response. So the acquisition of this HER2 gatekeeper mutation to us was really a telltale sign that these HER2 mutations are true driver mutations. So this cancer was so dependent on that HER2 mutation that in order to keep growing, it needed to mutate HER2 again so that HER2 was no longer blocked. So the SUMMIT trial, one of the earlier versions of the SUMMIT trial really showed that HER2 mutations are targetable, actionable mutations in metastatic breast cancer. So these are heavily pretreated metastatic breast cancer patients. And in particular, the combination of neratinib and fulvestrant in ER-positive HER2 mutant metastatic breast cancer was effective in at least a subset of these patients. The SUMMIT trial also showed that in addition to the acquisition of the gatekeeper mutation, something else that was really common at the time of progression was acquisition of mutations of a whole bunch of other mutations in the same HER2 gene. So this really seemed to be the dominant mechanism of resistance to neratinib in HER2 mutant breast cancer. And interestingly, many of these acquired HER2 mutations were sensitive to neratinib by themselves, but then they're popping up together with the original mutation. So these double mutations are popping up at the time of resistance. So we hypothesized that acquired secondary mutations in HER2 promote resistance to neratinib by enhancing HER2 activation and signaling and or by impacting drug binding. And this is the dissertation work of Arnaldo Marin. So Arnaldo systematically tested these acquired double HER2 mutations found in patients progressing on neratinib. And he found a few double mutations in particular that really shifted the IC50 and strongly reduced sensitivity to neratinib. And in addition, those same mutations strongly enhanced phosphorylation of downstream signaling targets and activated both the PI3 kinase AKT and MEK-ERK pathways. Now, computational structural modeling also showed that these HER2 double mutations stabilize the active conformation of HER2, leading to enhanced HER2 kinase activation, and they also impacted neratinib binding. So there are about 10 HER2 small molecule inhibitors in clinical trials, and Arnaldo tested them against the original HER2 L869R primary mutation, these acquired double mutations, and also the gatekeeper mutation. And what he found was that the cells with the acquired HER2 double mutations in L755S and T862A, they really showed reduced sensitivity to all of these HER2 inhibitors, suggesting that you really need a higher concentration of inhibitor to block that higher kinase activation with the double HER2 mutations. Now, in contrast, cells that expressed the gatekeeper mutation were resistant to neratinib, but they were still sensitive to the smaller inhibitors that were still able to fit within that binding pocket. Now, for these HER2 double mutations, if we can't treat them with other HER2 inhibitors, then what can we use? So Arnaldo tried combining neratinib with inhibitors of those downstream pathways. And he saw that combining neratinib with PI3 kinase or AKT inhibitors didn't really work that well. But combining neratinib with MEK inhibitors actually was very effective in cells expressing these double mutations, both in 2D and in 3D. So this suggests that a potential therapeutic strategy of combining a HER2 and MEK inhibitor in tumors that have these acquired HER2 double mutations. So now the current version of the SUMMIT trial tested the combination of neratinib, fulvestrant, and trastuzumab for HER2 mutant ER-positive metastatic breast cancer. And this trial actually led to the NCCN recommending this combination for HER2 mutant metastatic breast cancers following treatment with CDK4/6 inhibitors. So all the patients on this trial had previously been treated with CDK4/6 inhibitors and endocrine therapy. I should also mention that this was a randomized trial. The other two arms were fulvestrant alone or fulvestrant and trastuzumab, and there were no responses in the other arms, suggesting that neratinib is really needed to have these responses, that these tumors you really need to block that HER2 mutation with a kinase inhibitor. So about half the patients on this trial had ILC, and the responses were similar between ILC and IDC. Specifically for ILC, the overall response rate was 41% with a clinical benefit rate of 52%. And remember, these are heavily pretreated patients. The median progression-free survival was 8.3 months with a median duration of response of 14.4 months. But there were some patients on this trial that responded for several years. So this is a signal that, okay, it's a starting point, right? We have something that is now effective, that is actually targeting the driver mutation in these tumors. And we can hopefully build on this and make it better. Now, also, we saw that at the time of progression, even on this triplet therapy, still acquired mutations in HER2 were seen, suggesting that these resistant tumors still remain strongly dependent on that original mutant HER2 oncogene. So now I'd like to switch gears a little bit to early stage breast cancer. So we know, for example, for HER2 positive breast cancers, we know that HER2 inhibitors are effective in the metastatic setting, but they're rarely curative. Whereas HER2 inhibitors in early stage HER2 positive breast cancers, they have potential to prevent metastasis in some patients. And we also know that primary ILCs with HER2 mutations have a higher risk of recurrence. So can we do something about that? So will anti-HER2 therapy in early stage HER2 mutant ILC reduce recurrence? About 7 to 10% of primary ILCs have these activating HER2 mutations. And ILCs have very low rates of pathologic complete response to chemotherapy or endocrine therapy. So there is room for better treatments in the early stage. So we hypothesized that the combination of neratinib and endocrine therapy will improve clinical and molecular activity in early stage HER2 mutant ER-positive ILC. So together with Carlos Arteaga and Nisha Uni at UT Southwestern Medical Center and Laura Kennedy at Vanderbilt University Medical Center, we're planning a clinical trial for stage 1 to 3 hormone receptor positive ILC with HER2 mutations. Now, to find enough patients with HER2 mutations, we need to do genomic testing or next-gen sequencing on the diagnostic biopsy from about 400 patients to get 30 patients to enroll on our trial. So it is a big trial, but we did recently get an R01 to do this, so we're very excited about this. So the HER2 mutant patients will then be randomized to receive either neoadjuvant endocrine therapy, an aromatase inhibitor by itself for 4 weeks, or the combination of neoadjuvant endocrine therapy plus the HER2 inhibitor neratinib for 4 weeks, at which time we'll do a biopsy to see if the combination is better at promoting cell cycle arrest compared to endocrine therapy alone. And we'll also see if the combination can block phosphorylation of HER2 and those downstream targets. So then after these initial 4 weeks, all patients will then be treated with the combination of endocrine therapy and neratinib for an additional 20 weeks, followed by surgery. And our primary hypothesis is that the combination of neratinib and endocrine therapy will promote a lower PEPI score compared to historical controls of endocrine therapy alone. And a PEPI score is basically a surrogate for relapse-free survival. So if the endpoint is met, in addition to suggesting a new neoadjuvant therapy for HER2 mutant ILC, this would also suggest the value of next-gen sequencing or genomic testing in patients with newly diagnosed hormone receptor positive ILC, because right now, almost nobody with primary newly diagnosed breast cancer undergoes next-gen sequencing. Now, something else we're going to do from this trial is we are going to take fresh tissue from the patients to generate patient-derived organoids. So why organoids? Well, one limitation we've had with studying HER2 mutations in breast cancer and in ILC is a lack of patient-derived models with naturally occurring mutations in HER2. So we really want to build up that resource. In the meantime, we've relied on 2 PDXs, so patient-derived xenografts. One is from a ductal carcinoma and one is from an ILC. Both have HER2 mutations, and they are sensitive to either neratinib by itself or the combination of neratinib and fulvestrant. So we dissociated these xenografts to form these 3D organoid cultures, and we found that the histology of the organoids, or the PDX-derived organoids, PDXOs, recapitulates the histology that we see in the tumors. So they retain the lobular or ductal histology. And importantly, we also see that the PDX-derived organoids recapitulate the drug response that we saw in vivo. So our primary objective for using these HER2 mutant lobular organoids is to understand the evolution of response and resistance to the combination of estrogen deprivation and HER2 inhibition in HER2 mutant breast cancer to find ways to prevent that resistance from happening. So what about the other 90% of ILCs that don't have HER2 mutations? Well, it turns out that about 2/3 of primary ILCs have potentially actionable alterations. So one could envision a neoadjuvant umbrella trial, sort of like the neoadjuvant neratinib trial that I just showed you, that would start with next-gen sequencing, but then incorporate all these different alterations and match the alterations to targeted therapies to see if the combination of those therapies with endocrine therapy might be better than endocrine therapy alone. So I think this is really a great opportunity for more precise and effective treatments in the early stage that may prevent metastatic recurrence without needing to disrupt standard of care. So with that, I'd like to thank everybody who contributed to this. And I promised my kids that I would tell you all that they say hi. I'm not sure if you can see that. All right, thanks.
Thank you very much. Beautiful talk. We have time for some questions.
Thank you for your talk. A question for the design of the trial. Why wouldn't you try to add trastuzumab deruxtecan, knowing that the observation made for lung cancer with HER2 mutations?
Yeah, that's a great question. So the clinical data in breast cancer really isn't there yet for trastuzumab deruxtecan, specifically for the HER2 mutation. So we have the clinical data of neratinib being effective in the HER2 mutant metastatic breast cancer population, and we just don't have that data specifically for this cohort yet in breast cancer. Also, our models for trastuzumab deruxtecan, for our HER2 mutant models, it seems to be hit or miss. Sometimes it works, sometimes it doesn't, and we've seen more consistent results with HER2 tyrosine kinase inhibitors.
Okay, I have one question for you. So maybe a very naive question because I'm not a clinician, but looking at the melanoma field, if you do these types of inhibitions, you very effectively debulk for the one that is dependent on it, and then within half a year or maybe a bit later, people come back with other mutations because they're oncogene dependent on the same pathway. So aren't you afraid that if you do early breast cancer, you might pre-select for something that is just going to grow out again and is not going to be targetable by your drugs anymore?
I think that is a possibility. I think we're coming at it thinking that at that time, so after surgery, the overall tumor burden is going to be much lower. So hopefully, with a more effective therapy, we'll be able to get rid of those remaining tumor cells without having a large population with the risk that one of those tumor cells is going to acquire that mutation. But we don't know for sure.
Okay, thanks. Makes sense. Okay, thank you again. Round of applause for Ariella. So the final speaker of this session is Rinat Jeselson. She's from Dana-Farber Cancer Institute. Rinat is a medical oncologist who, just like the first speaker, holds an academic degree from the University of Jerusalem. Am I correct? Okay, she specializes on therapy resistance to estrogen receptor antagonists and cell cycle inhibitors in ER-positive breast cancer. And today she's going to present data on how a unique chromatin state drives therapeutic resistance in ILC.
So thank you first to the organizers. And can you hear me? Okay. For the invitation to talk here today and show some of our studies that we've been doing in lobular breast cancer. So as we all know, lobular breast cancer is a unique breast cancer. We know now that lobular breast cancer has, it's not working, has unique risk factors, or the risk factors are a little bit different, has a unique clinical presentation, unique histology, as well as unique percentages of the molecular subtypes, and also several studies have now shown unique genetic landscapes. But one question that remains is, does lobular breast cancers have a unique epigenetic state? And obviously that is a key question, since epigenetics, we know, is a key hallmark of cancer. And specifically, we were interested in understanding, does lobular breast cancers, do they have unique chromatin accessible sites? And is it different from invasive ductal cancers? So there is some data to suggest already that potentially lobular breast cancers have a unique chromatin state. And when you look at the genetic landscapes of lobular cancers versus ductal cancers, we now see from several studies that lobular breast cancers have an increased prevalence of FOXA1 mutations, with lobular cancers prevalence of FOXA1 mutations being 7 to 9% versus 2% in ductal cancers. And I know now they are called NST, but I will still call them IDCs. So what is FOXA1? FOXA1 is a key pioneer transcription factor that what it does, basically it binds to condensed chromatin, and then it displaces the histone 1 linker and enables the binding of other key transcription factors. And work done by Miles Brown's group many years ago showed that FOXA1 is required for most of ER binding, and really dictates estrogen receptor binding in ER-positive models. So to start to study the chromatin state of invasive lobular cancers, we turn to invasive lobular cancer cell models and as comparison to invasive ductal cancer cells. And this work was done by a really dedicated and talented group of researchers in my group. Agostina Nardone did all the wet lab experiments, and Shintao and Avery did the computational analyses. So the first experiment we did was we first did ATAC sequencing, which enables us to see the genome-wide chromatin accessible sites. And as you can see here in this sample-to-sample correlation plot, what we saw was that clearly the lobular models segregated from the ductal models, and this was due to close to more than 11,000 chromatin accessible sites that were gained in the lobular models compared to the ductal models. And also there were more than 5,000 sites that were gained in the ductal models. And when we looked at what are the motifs that are enriched in those sites that were gained in the lobular model, so the motifs are basically enables us to gives us some idea of what are the putative transcription factors that bind to these sites. And as you can see here, clearly the motif that was enriched in the gain sites in the lobular cells was FOXA1. So to see if this has any clinical relevance, we turn to the pan-cancer ATAC-seq study, which looked at ATAC-seq from the TCGA cohort. And this included 58 primary ER-positive breast cancers, including 38 IDCs and 14 invasive lobular cancers. And when we took this data and we looked specifically at our accessible sites that were gained in our lobular cell lines, what we saw was that in these tumors, we could find 3 clusters. One cluster, as highlighted in blue, is the cluster of tumors that were enriched in lobular cancers. So 11 out of the 14 lobular cancers were within that cluster. We identified a second cluster that was almost nearly all invasive ductal cancers. And then we saw a small cluster of tumors that did not have any lobular cancers and was also low in FOXA1 expression as well as ER expression and biologically more consistent with a triple negative breast cancer. So when we look at this cluster that has ILC, these ILC gain sites from our cell lines were also very tightly clustered very tightly and not shown here, but they also had increased accessibility in these specific sites, giving some evidence for the clinical relevance of our findings from our models. So because we saw that FOXA1 was the transcription factor enriched in these accessible sites, we performed FOXA1 chip sequencing in these models. And similar to the ATAC-seq through the FOXA1 system, we saw clustering of the ductal models from the lobular models, and this was due to more than 12,000 FOXA1 binding sites that were gained in the lobular models. And in this study, we did not see any FOXA1 ductal sites gained compared to lobular. And when we compared our ATAC-seq data to our FOXA1 chip data, it was really striking that all the sites that had gained accessible sites based on the ATAC-seq data overlapped with gained FOXA1 gain sites. So, of course, FOXA1 dictates ER binding. So we looked at ER chip, and very similarly, we saw that the ER system segregated from the ductal system, and lobular cancers segregated from the ductal system. And again, we saw binding sites that were uniquely gained in lobular cancers. And as shown in this enrichment plot to the right, when we compared FOXA1 chip to ER chip, we saw that those sites that had gained ER binding in the lobular breast cancers also had enriched FOXA1 chip sequencing. So, of course, chromatin accessibility, FOXA1 chip sequencing, ER sequencing all points to the fact that likely the transcriptomes of lobular cancers are very different from those of ductal cancers. And indeed, when we did RNA-seq, we saw that lobular models were different from the ductal models. And furthermore, when we looked at what are the genes that are regulated by estrogen, shown here in this dot plot, we saw that there was a very distinct set of genes that were estrogen upregulated in lobular cells versus those in ductal cells. And this was really consistent with work from Matthew Sikora from several years ago that he did when he was in Steffi's lab. So we next integrated our chip sequencing, our FOXA1 chip sequencing data with our RNA sequencing to really identify what are the genes that are directly mediated by FOXA1. And for this, we use a tool called Beta. And as you can see here, first in this volcano plot, what we're showing is basically when we look at the genes that are differentially expressed between lobular and ductal using our RNA-seq. And then we look at the genes that are putatively regulated by FOXA1 uniquely in lobular cells based on our FOXA1 chip sequencing data. We saw that there was a strong, significant correlation between these genes. And as you can see in this plot, is that the genes that were putative downstream to FOXA1 binding were all upregulated in the lobular models. We also use beta to integrate this data to identify those genes that are direct FOXA1 regulated genes, integrating both the RNA-seq and the CHIP data. And by integrating the FOXA1 gain sites and the RNA-seq looking at comparing lobular to ductal, we found a signature of genes. And you can see that these genes are enriched in pathways of, not surprisingly, ER signaling, but also other genes that are important in tumor progression, such as Notch signaling, TGF-beta, and hypoxia. And when we narrowed this gene list using a very stringent statistical cutoff, we came to a gene list of 120 genes. So this is not from P120, but this is happened to be 120 very significant genes. And we can see when we use those genes and we turn to the MetaBrick cohort and we compared ILC to IDC, we see indeed that these genes are upregulated in the ILC tumors. And moreover, when we looked at outcomes, we saw that this gene signature was associated with worse outcomes, specifically in ILC tumors that were of luminal A subtype. This is important because this gene signature could potentially help us identify those tumors, ILC tumors, that based on their PAM50 potentially have good outcomes, but actually have poor outcomes. In addition, it's important to note that this signature was not associated with outcomes in luminal B breast cancers that are, we know they're less driven by ER signaling, and they have other genetic alterations that likely account for their poor outcome, worse outcomes. So this led us to ask, what is driving the FOXA1 reprogramming in ILC? So from previous studies, we know that FOXA1 mutations and FOXA1 amplifications are associated both with increased FOXA1 expression and also with the reprogramming of FOXA1. And when we looked in our cell line models, we performed exome sequencing. We did not identify FOXA1 alterations in these cells. But what we did see was that there was FOXA1 overexpression, and this was both at the transcriptional level and at the protein level. And that led us to ask what regulates FOXA1 expression. So for that, we turned to the SystemDB toolkit. This is a publicly available website that has curated chip sequencing experiments from thousands, maybe thousands by now, of chip sequencing published data. And what we saw was that the top putative regulator of FOXA1 is FOXA1 itself, which is not surprising because many transcription factors are autoregulated. And that led us to go back to our chip sequencing data, to our FOXA1 chip sequencing data, to look specifically at the FOXA1 gene site. And surprising to us, what we found was that upstream to FOXA1, there was a strong peak that was unique to our lobular models. In addition, we saw that this same peak was found in primary lobular breast cancer cells that we isolated from metastatic acidic fluid. And when we looked at that site and we looked at our H3K27 acetylation data, we saw that this site overlapped with a super enhancer only in our lobular models. And furthermore, this site also was enriched and had a unique lobular unique ER and GATA3 binding sites, further substantiating the role of this site as a super enhancer, since super enhancers are known to have a binding of multiple transcription factors that are lineage defining. So to functionally test what the role of this peak that we identified, we performed CRISPR-I specifically targeting this peak. And you can see here that when we targeted this peak, we saw a decrease of FOXA1 expression at the protein level. We saw a decrease of FOXA1 in the transcriptional level, as well as other genes that are FOXA1 regulated, such as ESR1, and genes that are ER regulated. But importantly, we saw that the targeting of this peak led to significant repression of growth in lobular cells. And as controls, when we looked at MCF7 cells and we validated that this guide that the DCAS9 crab is working in this model, we saw that with targeting this peak, we didn't see an effect in MCF7 cells. So to summarize, what I've shown you is that ILC has a unique chromatin state that is tightly linked to a reprogrammed FOXA1 sistrum. The ILC unique FOXA1 sistrum is associated with a unique ER sistrum and ILC unique response to estrogen. I didn't show you this, but in our published paper, we do show that it also is associated with, it also provides a mechanism to decrease response to tamoxifen. In addition, we identified a FOXA1 ILC 120 gene signature that is associated with poor long-term outcomes in luminal A ILC. And we also show a proof of concept of precise targeting of an ILC unique FOXA1 peak upstream to the FOXA1 gene that resulted in significant suppression of cell growth, offering a potential therapeutic approach, although there's still ways to come to identify a small molecule that can really do that. So we are currently continuing this work and really trying to shift more to the clinical side and see if we find similar findings in clinical samples. And I'm very fortunate that to work with my colleague Otto Metzger, who conducted a clinical trial, which was one of the first clinical trials to be really enriched in lobular breast cancer patients. So this is a neoadjuvant clinical trial that was enriched for patients with lobular breast cancer and randomized patients to tamoxifen versus letrozole initially, and then to letrozole plus palbociclib versus letrozole. This trial has completed accrual. It accrued 189 patients. And indeed, we really enriched for patients with invasive lobular breast cancers. About close to 50% of the patients had invasive lobular cancer in this trial. So tumor tissues were collected at baseline after 2 weeks and at the time of surgery. And we are now really working very hard on the analysis of these samples. In addition, as part of this work, very grateful to the LBCA for granting postdoc in my lab, Capucina Rhoda, a grant to perform multi-omic analysis on samples from this study. So our plan is to do single-cell RNA-seq and single-cell ATAC-seq on ILC and IDC tumors, pre-treatment and post-treatment, to really see if the findings we found in our cells really pan out in patient samples, and does it have therapeutic implications. So with this, I will end and thank the people in my lab, my collaborators, and funding resources. Thank you.
Thank you very much. Your timing is impeccable, so we have time for questions. Anyone?
Hi, thank you for a great talk. This is Robin from Baylor College of Medicine. I'm pretty new to this field, so I have two naive questions. Sure. The first is for the IDC and ILC, both in cell lines and the patient primary tumors you look at. Are there any differences in terms of the expression level of estrogen receptor?
So in general, ILC is high ER expression. The ILC models that we use have very high ER expression as well.
Thank you. So sorry, So another question is, do you have any hypothesis on why is there a distinct FOXA1 like different from IDC? ILC versus IDC differences?
Yeah, I mean, so one thing we haven't solved everything, and one question is always what comes first? Is it the chromatin state, or is it the FOXA1 driving the chromatin state? But just because of the way FOXA1 works, it probably is. Potentially, it is FOXA1. We know from other studies that an overexpression of a transcription factor or can change the reprogramming of that transcription factor. So that's our hypothesis. It's a little hard to tease that out. We did do FOXA1 silencing, and we did see that by silencing FOXA1, there was a big shift in the chromatin state. It wasn't 100%, but we do see a pretty significant change.
Thanks for the talk. Can I ask, related note, do you think E-Curvin loss in maybe NSTs, would it reprogram the FOXA1 landscape? Do you think if you knock down E-Cadherin, would that result in the same landscape? So is E-Cadherin the event that results in this, I guess, epigenetic change? Or do you think this change happens first, and then maybe E-Cadherin is the next step?
Yeah, so I didn't show it here, but we also did silencing of E-Cadherin in the IDC models to see if that affect the H3K27 acetylation or global RNA-seq. And we saw very little changes in transcription. I know Steffi, I think, did similar experiments. We also overexpressed E-Cadherin back in the ILC cells like somebody else did here. And we looked at transcription and we saw very few differences. Most of the genes that we saw that were related really to cell-cell contact. So I think this is probably a separate from E-Cadherin, but I think it does highlight the importance of the ER axis being different in ILC compared to IDC, and it's really not just E-Cadherin loss.
Rinath, can I just follow on from those questions because I think they're kind of interesting. So then do you think if the reprogramming came before the mutation, and the mutation is a random event, do you think normal lobular cells of the breast have a different chromatin, like FOXA1 chromatin state? Is that known?
So
compared to normal ductal cells, yeah.
If it's in lobular,
like lobular cells, do they have a distinct chromatin state that then is different?
Oh yeah, I don't know. Like, yeah, it would be nice if we could find the cell of origin to see if that's really there. Yeah.
Okay, if there are no more questions, then let's have another round of applause for the speakers.